This journey started when we got to know about planarian flatworms through our colleague Dasaradhi. Planarians are remarkable organisms that can fully regenerate. You can cut these worms into tiny little pieces, and if you just wait for a few days, each piece will regenerate to give you back a complete organism. While a lot of focus has been on planarian regeneration, we decided to focus on function. What do these worms actually do? What are they capable of? And what started as some simple, curiosity-driven experiments on light sensing by my student Nishan and others led to a series of frankly eye-opening discoveries. Planarians are highly light aversive, sensing light through simple cup-shaped eyes linked to a brain. Such simple eyes are fairly common throughout the natural world. Scientists going all the way back to Darwin have commented on the evolutionary significance of simple eyes, but much less is known on what these eyes actually accomplish. Planarians also happen to have one of the first examples of a brain in evolution, making our findings all the more exciting. Our research shows that planarian light sensing is surprisingly sophisticated. You can give the worms two very similar light inputs and they are able to compare differences between the two and give virtually the same result every single time. Quite simply, they are ultra sensors with comparative processing, where they can sense even small changes in light and are able to completely switch their behavior accordingly. Now this is where regeneration comes in handy. If we cut away their heads, they of course cannot sense visible light. But these are planarians after all, so over time they will regenerate their eyes and their brain. During regeneration, if we time it just right, we find worms that can sense light. They are not blind, but they cannot compare small differences. Then, as their eyes and brain strengthen, they build the right neural connections and their abilities recover. So essentially, we can watch the recovery of comparative processing, which is quite unique. This now allows us to link eye-brain regeneration to recovery of function. Maybe we can even dare to ask as to how these worms compute and compare. This story has another twist. These worms can also sense light without eyes. If we shine even tiny amounts of long UV light, headless worms start moving like intact worms and show an escape response. This reflex-like response is quite different from eye-brain sensing. So we have two amazing but very different light sensing networks in one organism, allowing us to do really unique experiments. We were even able to challenge the worms with choices and ask which neural network would win, the reflex-like whole-body network or the brain-based network capable of processing. We found that in intact worms, the eye-brain response can override the whole-body response. This is quite striking and is reminiscent of hierarchies in neural networks in more complex animals and mammals. Studying eye-brain regeneration showed that these hierarchies in planarians are quite dynamic. There are some time points where the whole-body response is actually stronger, not yet under the control of the newly forming eye and brain. Then, in a clear, sharp switch, the eye-brain network reclaims its dominance. This switching depends on how regenerating neural networks rewire and reconnect to each other. The exciting thing is, we now have a method to visualize this rewiring and competition between two very different neural networks, all in the same animal. In summary, for these worms, light truly matters. They have evolved the ability to process light in multiple different yet sophisticated ways using seemingly simple apparatus, as they must have adapted to their complex natural habitats. Here we learn so much about natural light sensing as well as neural regeneration by bringing a fresh new perspective to a fascinating model organism. This illustrates as to how curiosity-driven research not only leads to new discoveries about the natural world, but also can impact problems like neural regeneration that have tremendous biomedical relevance.